This is the second video for our Chapter 9 week. We've already talked about Mendelian genetics and the basis of single gene heritability. Now we're going to talk about a few variations of the basics that Mendel discovered. As we have been doing, initially we're going to go over some important words, then we will talk about pedigree, human genetic disorders, a few examples. There are many, obviously, but we'll talk about a few common ones that are rare in that there is only one gene that causes them. We'll briefly touch on the problem with inbreeding in a genetic term. We'll move on to heritable variations. Instead of single loci simple dominance, like in Mendelian genetics, we'll go into incomplete dominance, codominance, and pleiotropy. We're going to touch on nature versus nurture, genes that are linked, what genes exist on the chromosomes that are sex-determining chromosomes, because those aren't only, the genes on those chromosomes don't only become involved with sexual reproductive organs and processes. They're involved in many other things as well. Okay, so here we will touch on vocabulary. A term that is used in your book frequently is wild type. And the basis of understanding this term is that not all dominant genes are more common. The wild type is basically a way to reference the genetic combination that occurs regularly or most frequently in a particular population. Next is chromosomal basis of inheritance, and this is pretty simple. Obviously we know that genes are on chromosomes which are inherited from your parents, and these contain the blueprints for how to make an organism. But the basis of chromosomal inheritance is that during meiosis, the way genes line up and have crossed over determines how they will be passed on to the next generation. Incomplete dominance is something we will talk about where, say you have a dominant allele coding for a gene and a recessive allele coding for a gene in the individual. Incomplete dominance occurs when that dominant allele isn't completely expressed as a dominant feature. When you end up with something in between, it's called incomplete dominance. Like you have flowers that are red that breed with flowers that are white, and then your F1 generation turns out pink. That would be incomplete dominance. Where if it were regular complete dominance, the offspring would be, say, red would be the dominant color. All the offspring would be red. So another variation of inheritable genes is codominant, when basically there can be several genes that come into play in expressing a particular trait or characteristic, and this is when all traits are expressed. All traits that are present are expressed. There isn't a very simple definition between what is dominant and what is recessive. So it's different from incomplete dominance, where in incomplete dominance there's a gray area between each gene, and what the phenotype is expressed somewhere along that gray area. In codominance, and we'll talk about this with blood types, each gene is expressed, or each allele is expressed, and we'll go into some examples. Okay, and then there's pleiotropy. And what that's referring to is where, there's a typo there, the control of more than one phenotypic character by a single gene. So when a single gene codes for different phenotypic expressions, so one gene controls several different ways or several different expressions in an organism. We'll have a small example on that as well. So now we're going to touch on pedigree. I believe someone in class asked if you display or express the characteristic of a dominant allele, how do you know whether you are homozygous or heterozygous? I mean, short of going in and having your blood tested and all your genes sequenced, what researchers have done historically is something called a pedigree. And what that essentially is a family tree. When you have an individual whose phenotype you know, say they express the recessive trait of some characteristic. You can trace that back, looking at their relatives in the current familial structure, looking at their parents, looking at their parents' brothers and sisters, looking at their parents' parents, and hopefully by constructing a chart of the traits that are expressed in their family members, you can try to figure out what their genotype is or how a particular trait is passed on through their family. And again, this is where we'll use the term wild type, where a phenotype that is more common does not necessarily mean that it's dominant. And in light of that, it's good to keep in mind that very, very few traits that we have follow the rules that we saw in Mendel's experiment. Very few genes apply to the laws of simple dominance on a single gene. Most things that we express are coded for on many different gene loci. So there are many genes that come into play to create how your body expresses your genetic code. Like hair color isn't determined by a single gene on your chromosomes. It's determined by a series of genes, potentially on different chromosomes, that have to work in concert or together, generating the proper proteins that will express your hair color. And it'll change over time. So it's not simply that if your dad has black hair and your mom has blonde hair that you're going to have dark hair. You might have some variation in between, and it's not determined by just one specific location. Okay, and so this is an example where maybe it'll be easier to understand wild type genes, or just to see visually that yes, often recessive genetic combinations can be more dominant in a population. Because if you look at the three, or at least the first two pictures on the top, I think it's more common that we see people who don't have freckles than people who do, even though some of the genes that come into play for freckles are dominant. Same with widow's peak on the hairline. It's more common that you see people who do not have a widow's peak than people who do. Now, as far as earlobes, I don't know, the general ratio in the populace, in like human populations, whether more people have free earlobes or attached earlobes, but the example that you need to take away from this particular picture is that it's not guaranteed that wild type traits are dominant traits. Oftentimes, dominant traits are seen less frequently in a population. But what we were talking about is pedigree. So I want to show you just a very, very small example, a short example of how someone has done a pedigree, and this is something relatively simple. First, I want you to look at the key in the bottom left here, where circles represent females, squares represent males on this particular pedigree, and the filled in shapes represent someone who has attached earlobe, and the empty shapes represent someone who has free earlobes. And as we know, free earlobes are dominant, these are recessive. If it's only one gene that determines this, you already know the genotype of the individual who has attached earlobes because it's recessive. Looking at the family tree, say you start with what you know. Kevin is a male and has attached earlobes. So you know for certain that his genotype is FF, based on his phenotype, which is the expression of attached earlobes. Now, Kevin's sister Lisa does not have attached earlobes, so that is the dominant trait, meaning that she could be homozygous for the dominant trait or heterozygous for the dominant trait. And family trees you have to follow backwards, just because that's the easiest way to do it. I guess you don't have to, but that's the natural way that you would go. You start with what you know and who you know and work from there. So if we take Kevin and Lisa and we look at their parents, Hal and Ina. Hal and Ina both do not have attached earlobe. So the next step would be to look at the siblings of these two individuals, where looking at Ina, Ina's got a sister. Her sister is Jill, and Jill has attached earlobes, so she's purple, and you know that her genotype is recessive. Now how do you think you know what Kevin and Lisa's parent genotype is? You see that it's been put down here as heterozygous? You know that because Kevin exists and has the recessive trait. He had to get one from each parent, so that means Hal is heterozygous because he's got to have a recessive trait to give to Kevin, as is Ina. Ina is also heterozygous because each parent has to have a recessive allele to give to this individual. So we know their genotype as well. We still don't know Lisa's, but we know these for sure. Now, still looking at Ina's family, we're going to go back to Ina's family. So Ina is definitely heterozygous and Jill is definitely homozygous recessive. So let's take a step up and look at their parents. And it looks like Ina and Jill's father, Cletus, also had attached earlobes. So his genotype is easy to identify as homozygous recessive, and then you know that their mother has to be heterozygous because Jill expresses the recessive trait, and she had to get a recessive trait from each parent. So Deborah definitely had one recessive allele, and because they have a daughter, Ina, who does not express the recessive trait, Deborah has to be heterozygous as well, because somehow Ina had to get a dominant allele, so it came from Deborah. Okay, now we're going to move over and look at Hal's family. Hal had 3 siblings, 4 children in total. Looks like 2 brothers with attached earlobes, so their genotype is easy to determine, both homozygous recessive, and then a sister who does not have attached earlobes. Now, we can't determine her genotype right away, so let's go up and look at Hal's parents. Now, neither of Hal's parents, Aaron or Betty, had attached earlobes, but because Fred and Gabe did, that means Aaron and Betty each had to carry a recessive allele. So you know that they are heterozygous. Leaving Evelyn. Evelyn does not have attached earlobes, but we can't determine for sure whether she is homozygous or heterozygous, unless maybe if she has children, if she ends up having a child, potentially with the recessive allele with the attached earlobes, then we would know that she were heterozygous, but if she doesn't, we really have no way to tell the difference. So I hope you see now how it's easy to use a pedigree to try to determine the genotype of individuals, and this is important, especially when a particular trait is dangerous or can have negative effects on an individual, to try to trace if someone's offspring have a chance of inheriting an allele that's a little less benign than simply earlobes, maybe an allele that causes a disease that you don't want, and we're going to talk about that here in a minute. So this is a pedigree. This is something that's been used for generations to try to determine how genes are passed through families. Now, we've mentioned genes that may have negative effects on individuals. This is a chart of human disorders or diseases that are caused by a single gene. Some are recessive, some are dominant, and they do tend to happen with increased frequency in particular populations. If you look up over here in incidence, they don't have albinism as being correlated to any particular type of people, so maybe it's got the same, it occurs at the same rate within all human beings. But cystic fibrosis, you have a 1 in 1,800 chance, or 1 in every 1,800 European Americans carries a gene for cystic fibrosis, or at least expresses the disorder in European Americans. So that means the original mutation that occurred in this population, like sickle cell disease, is very common in African Americans. So that particular genetic variation happened. It's a simple mutation. We're all mutants in one way or another. That happens in African populations, 1 in 500. So let's just look at the list instead of looking at incidence. Albinism is pretty simple, lack of pigment. I think we're all familiar with that. Cystic fibrosis is when an individual's body, there's an enzyme, a protein, whose blueprint is a little screwed up, and what happens is the signaling in the body ends up making more mucus than it needs, which leads to decreased function in an individual. The PKU disorder leads to an accumulation of phenylalanine in the blood, lack of normal skin pigment, and mental retardation unless the diet has been changed, because what happens with that, it's diet specific, but if an individual continues to eat particular items that their body can't handle, then, well, basically their body can't break it down, so they get an accumulation of phenylalanine, which inhibits brain function or can cause delay in proper development of mental capabilities. So once the diet's been changed, that individual will be fine. But it's about catching it and figuring out. You got to know the symptoms, and there are many symptoms that with PKU, like the smell of somebody's urine and other things to keep an eye out for when an individual is an infant. We've already talked a little about sickle cell disease, and that's a protein change shape in the hemoglobin, which are your blood cells. And the reason it's so common, even though it can be deleterious or it can cause damage to an individual's systems if they are homozygous for the gene. If they're heterozygous, they can still function relatively well, but it has an evolutionary advantage in that there is a disease called malaria that I'm sure you've heard of, that sickle cell will help somebody survive because the malarial parasite cannot survive in the sickled blood cells. And that's part of the malarial parasite's life cycle. Then there's Tay-Sachs disease, where the body can't handle lipids appropriately, so they accumulate in an individual's body, decreasing mental capabilities. And generally, I believe these children don't live beyond 5 years old. And it happens most commonly in European Jewish populations, probably because that's where the mutation first occurred. You never know when these genes are when your genes or anybody's DNA molecules are going to be altered, and what's going to be passed on. It can happen completely randomly. So those are simply recessive disorders. So those disorders only come about when each parent is a carrier, when each parent is heterozygous, and any of their offspring may end up with both recessive alleles. Now, there are also dominant disorders. And as you may realize, many of these are not common in the population. Achondroplasia, I don't know if I'm saying that right, which is dwarfism, is a dominant allele, but it's not frequent. Only 1 in 25,000 people may have this. That one's pretty simple. You guys know about it. There's that TV show Family, which is actually in our book. So you've read about dwarfism. Okay, so there is a type of Alzheimer's disease, which is a single gene disorder that is actually dominant. And oftentimes, like I may have mentioned before, the difficulty with dominant deleterious diseases or disorders is that they do not show up until later in life. So an individual often doesn't have the opportunity to think about whether they want to pass this on to their offspring or not. And the next few examples of disorders can present a dilemma for individuals when it comes to thinking about passing these types of things on. So there's one type of Alzheimer's disease. I think most people are familiar with it now, where there is neural degeneration. It is a disease that strikes much later in life, generally in the elderly, and we know it is inherited, although there are many different forms. Some others that we're going to talk about, we're going to talk a lot about Huntington's disease, and this one is particularly insidious because an individual has no symptoms until mid to late 30s or 40s, and by then they have already had children and hadn't had the opportunity to consider whether they want to pass something like this on. Not that, it's an individual's choice. And Huntington's disease is something that's really very recently been thought about. So that's one that we're going to go into with more clarity or more in depth here in just a moment. And your book talks about hypercholesterolemia, where if an individual has the dominant version of this gene, they have a lot of cholesterol in their blood and may have heart attacks as early as the age of 30. And if you look at the incidence on the far right of this graph, 1 in 500 individuals carries this, which is relatively common. Moving on, we'll take a little bit of a closer look at cystic fibrosis and then a closer look at Huntington's disease. So cystic fibrosis is recessive. Since it is recessive, an individual doesn't generally know if they're a carrier. They only know if they end up with an offspring that expresses the symptoms of being excess mucus production. And excess mucus can get in the way of many, many other bodily functions, like breathing and digestion and absorption of nutrients. I've included a link at the bottom to a government website that has more information on each of these diseases. It's likely somebody in class knows someone who is affected or has one of these. So cystic fibrosis is relatively common. There are many organizations that are out to try to cure this particular disease. One thing I find interesting, though, is how do you cure a disease that's genetic in its origin? It's not like you can change all of the DNA in your body to alter the way that this particular gene is expressed, or maybe there are other ways to do that. There was once a successful treatment of 2 girls in 1990 who were cured of a rare blood disorder caused by a defective gene for the enzyme adenosine deaminase. There was a procedure called gene transfer therapy, where scientists isolated working copies of the gene and introduced them into bone marrow cells taken from the girls. The modified bone marrow was allowed to proliferate, and then the girls' own cells with this new genetic incorporation were injected back into their bodies. Both girls recovered from this blood disorder and stayed healthy. That's the first time in 1990 that a genetic disorder had been cured by gene therapy, but it can't always be successful. I think that's a really rare case, because oftentimes the way a body functions and all of its processes is a little more complicated, although there is a lot of money going into trying to cure cystic fibrosis, as well as the next one we're going to talk about, which is Huntington's disease. Cystic fibrosis is a little insidious, just like Huntington's disease, in the way that you don't know that you're passing it on, and it's recessive. That people can be carriers, many generations can be carriers before the disease actually surfaces, depending on who they choose as their mate. And it's not guaranteed. It's not like a 50-50 shot for the offspring. There's a 1 in 4 chance that an individual will have cystic fibrosis if both of their parents are heterozygous. Then there's Huntington's disease. This is a dominant gene, so any individual who has Huntington's disease has a 50% chance of passing it on to their offspring. It's very scary because, like I mentioned before, symptoms don't occur until an individual is in there between 30 and 50 years old. And by then, most people have already had children. And Huntington's disease, they didn't start looking for the particular gene for it until about 1983. They didn't find the gene until 1993. So it's just been recently that they even knew how to look for it or find it. So that means many people didn't really know the details about it in their family histories, and that leaves a difficult decision for individuals who were born between the 80s and 90s who now know that their parents have Huntington's disease, and it puts them in an interesting position to decide whether or not they are going to choose to potentially pass that on. A particularly famous case is Woody Guthrie. He died of Huntington's disease and had a son, Arlo, who has chosen not to have children just because at the time, now he didn't have a way to know for sure whether or not he carried the disease. There was no way to know, and that's a difficult choice to make. His mother and him have actually funded an incredible organization to do research into finding ways to ease the symptoms of Huntington's disease. Now, Huntington's disease is a single gene, and there's a location on it where the CAG nucleotides are repeated more so than is common in a normal gene for that code. And basically what eventually happens is once a person begins to be affected, their brain, their neural pathways literally start deteriorating. They may lose motor skills, but eventually they lose the ability to think and care for themselves, and this disease will eventually kill them. Not the disease itself, but they will die by some other way, usually choking or pneumonia or some other form. After the onset of the disease, people usually live 10, maybe 25 more years with this slow but certain deterioration. So moving on, we mentioned incomplete dominance. This is when you have alleles for two different traits, where each parent, this is looking at snapdragons, an individual snapdragon here codes for red, this one codes for yellow, and then if you cross the pollen from the white to the red plant, pollen onto the stigma, if you take the pollen from one of the flowers and put it on the stigma of the other, all of the offspring will be pink. And then if you do the Punnett square, the individuals that are homozygous for the red coding gene will be red. The offspring that are homozygous for the white coding gene will be white. This receives two red coding copies of the gene. This one receives two white. But the individuals that receive a red and a white turn out pink. So that is incomplete dominance is a blending of the characteristics of the gene. So it's not complete dominance and recessiveness. When each is coded, they both share in the expression of the individual. So next we're going to look at codominance, and this is where when different alleles are present, they are each expressed, and our blood types are the perfect example, where there are not just two different kinds of alleles here, there are actually three, where an individual can have the A, we're just going to call them what they are because they put different proteins on the individual blood cells, or none at all. So there's A, B, and O, which is no, codes for no identifying proteins on the outside of the hemoglobin, or carbohydrates, I'm sorry, carbohydrates. So if you have the A gene, you will express A carbohydrates, these are A's, on the outside of your blood cells, or B. If you have both A and B, then you will have both carbohydrates existing on the outside of your hemoglobin. And if you carry the gene that does not code for any exterior carbohydrates on your hemoglobin, you won't have any at all. That's type O, or in this particular case they use little I. So if an individual has both A or AI, or little I, big A, little I, genetic combinations, this is what their blood cells will look like. If an individual has B, or B and the O blood type, they will have the B carbohydrates on their blood cells. If their genotype codes for both A and B, they will express both, or if they're double O, they will have none. Now this is important because in blood transfusions, if an individual has type A blood and is given, say, type B blood, the blood will essentially all conglomerate or coagulate, and it will kill an individual. That's why blood typing is important, but what we're really just looking at is codominance here. What it means is that each gene that is carried by the individual is expressed in the phenotype. Now this is the graph from your book about sickle cell anemia. So when it's called Pleiotropy, that is when an allele may be dominant with respect to one phenotypic consequence and recessive with respect to another. This is where this is where one gene affects many traits in an individual. So they use sickle cell anemia as an example because in sickle cell anemia patients, a defect in the oxygen-carrying hemoglobin molecule causes anemia, heart failure, increased susceptibility to pneumonia, kidney failure, enlargement of the spleen, and many other symptoms. This one particular change in one gene causes many differences in the individual. It's also called pleiotropy because an individual, when they are homozygous for the trait, they have most of their red blood cells will sickle, and it's extremely difficult for them to even survive. But in a way, it's also incomplete dominance, or codominance, I'm not really sure, because a heterozygous individual will display some traits of sickle cell anemia, but will still be able to survive and produce offspring, where frequently an individual who is homozygous will not produce offspring, or generally will not live long enough to do so. But because that single gene has many effects within the body. It's called pleiotropy. Another example, there was a French scientist who was trying to get a particular yellow color of fur in mice. He knew it was dominant, but when he crossed the mice and would get a homozygous offspring, that offspring would typically die because that gene coded for, or another effect of that particular code for the gene caused something in the mice not to work properly, so they would die, but a heterozygous individual would survive. So next, your book talks about the difference between how we know genes code for many, many different things in your life, but there is always an environmental factor as well. They give you a picture of identical twins, and everybody, I think most of you probably know someone who is a twin, and as they grow older in age, or even when they're young, you can tell they are not exactly the same person. They each lead their own lives, and different environmental factors that come into play in their own lives will lead to differences in their genetic expression. But how do you determine how much is nature and how much is nurture? That is the ultimate conversation in determining, trying to tease out the differences between what really is a genetic effect of a disorder and what is not. So while genes do code for all of your traits, they cannot be used as the culprit for everything that happens in your life. A lot of things that happen are still environmentally influenced. A lot of traits that you may express are environmentally influenced. Okay. So we've talked about Mendel. You saw that in the other video where it assumes each gene is transferred independent of any other gene. So flower color was transferred independently of seed color, independently of pod shape, independently of plant size, and so on. And we were able to, look at how these genes are inherited individually because of the processes of crossing over and how the chromosomes align during meiosis. But there is another experiment that your book talks about that shows how some genes may actually be linked. Now, it comes into play where many traits that are expressed by an individual are not coded for on one specific location of a chromosome. Many times, one trait will be coded for by several different locations on a chromosome. And I hope that makes sense, where sometimes if those genetic locations are relatively far apart from each other on one individual chromosome, it's possible that during crossing over, some of those genes will be changed and the ultimate expression will be altered. Another thing that may happen is that it may be observed that many traits are transferred together. This graph here shows where more frequently this particular individual found offspring. When he crossed heterozygous with homozygous, the offspring would more frequently look just like the P1 generation. The F1 generation would exhibit similar traits to the P1. They did not follow the Mendelian Punnett square, the expected outcome. So the scientists came up with the idea of linked genes. Although it is possible that some of the crossing over occurrences happened right at where those genes were close to each other, thereby producing only a few of the offspring that would have a different combination of the genes. Now we're going to move to sex-linked genes. So what's important to understand here, of course, you've got X and Y, but the genes on these chromosomes do not only code for the differences between male and female, they code for other things as well. So as you know, in sexual reproduction, each offspring will get an X chromosome from their mother. The mother can only donate an X chromosome because that's all she's got. She's got double X's. And then the father can donate either an X or a Y chromosome. That's why the sex of a child is determined by which sperm fertilizes the egg. So what may happen, though, a lot of times, is that in a male individual, he cannot receive an X chromosome from his dad. the X chromosome that all boys have is from their mother, for sure. And the Y chromosome is from their dad. So if there is a gene loci on here that codes for something like colorblindness, it will be expressed in the male. This is why genes that show up on the X chromosome are more often phenotypically expressed in males than in females, because females have a secondary X chromosome where if that X has another gene on it, like say this little girl receives the colorblindness gene from her mom, but if her dad is not colorblind, she will not be. But if mom is a carrier and submits a colorblind gene to her and her dad is colorblind, then this little girl will be colorblind. But if dad is colorblind, mom is not, or no, dad is not colorblind but mom is a carrier, there's always a chance that a male offspring will receive the colorblind X from the mother. He cannot receive it from the father, so it's basically a sex-linked chromosome. This is just a brief rundown of sex-linked genes and why males more frequently express the characteristics of a defective gene that's on the X chromosome. It's because they have a Y chromosome. They do not have another X chromosome to potentially override the negative effects of any genes that are on their X chromosome. Women typically only express the phenotype of defective genes if they are homozygous on their X chromosomes, which happens relatively infrequently. Two of the most common sex-linked disorders or defective genes are colorblindness and hemophilia. Typically but most typically males are colorblind, and hemophilia is most frequently expressed in males as well. So this is a simple colorblindness test here. If you're colorblind, you can't see the number 7 in there because it's in green dots. Colorblindness only affects an individual's ability to differentiate red and green, mostly. There are different variations of colorblindness, but the most common is just the ability to see red and green colors differently. Normal female with a colorblind male, showing that any daughters they have will be carriers of colorblindness because dad only has one X gene to give to his daughters, and it will have the colorblindness gene on it, but none of their offspring will actually express it because if mom is homozygous, they will all receive a good copy of the vision gene, essentially. But if mom is heterozygous, there is a 50% chance that any male offspring will be colorblind, even if dad is not. So, and then a potential that they will have one girl who may be a carrier. And then if mom is a carrier and dad is colorblind, any girls that they may have will definitely receive one copy of the colorblind gene, and the males essentially have a 50-50 chance, because if mom is heterozygous, he could get a good gene, could get the colorblind gene. Now this is essentially what we went over: important words, what a pedigree is for humans, common genetic disorders, recessive versus dominant. Oh, and I forgot to talk about inbreeding. Basically, the issue with inbreeding is that only in genetic terms, when individuals that are more closely related mate, and if there is a defective gene that is recessive, it will appear more frequently in a population that breeds only within that small population, because there will be more individuals that are heterozygous, and the more individuals that are heterozygous that mate, the higher the frequency of individuals that will be homozygous for a recessive deleterious trait. Basically, the more frequently they will trade and pass on any defective genes. We talked about heritable variations, where not every genetic combination has simple dominance. Oftentimes, there will be expressions of some range between the genetic material, or where all alleles that are present will be expressed, or if one particular gene codes for the expression of many different types of traits. Nature versus nurture is a common, long, and ongoing conversation in biology where we still don't know how to exactly tease out the difference. There are linked genes that occur frequently very close to each other on a chromosome, so very frequently they are passed on together, although they're not 100% of the time. And then sex-linked genes, where males are more susceptible to any defective genes that are present on the X chromosome. Now that's only in humans. Many other species' sexes are determined by a different combination of genes.